All right, welcome everyone. Welcome to participating this event organized by SSE. This is Symposium, International Symposium for Mandarin speakers in East and Southeast Asia. On behalf of the organization, we're from the Society for the Study of Evolution and other organizations. I'm Chonghao Susu. I'm very happy to share with you, and I'm very happy to say hi to you in Mandarin, my mother language. Hopefully, we'll all have a very fruitful results and also have a wonderful exchange. First of all, I would like to thank the organizer. I would like to thank the SSC and each committee members and also each department experts. Thank you for your support and your assistance. This is also an event organized by SSE. I would also like to thank local organizers. I would like to thank local organizers from each institutes, universities, colleges. Thank you for our models and thank you for these experts. Thank you, everyone. Now, before I begin today's session, there are some housekeeping that I would like to share with you. First of all, this seminar will be recorded. You can use the Q&A module to ask questions. Click on the Q&A and you can submit your questions. Our presenters and speakers will respond to you in real time. Third thing is that for this seminar, it will be conducted in Chinese, but at the same time, we also provide you simultaneous interpretation. So on the interpretation function, you can actually click on the globe icon and select the language that you prefer to choose. Our simultaneous interpreters will translate in real time. Once again, thank you everyone for coming. And I would also like to thank all the local organizers and SSC for the wonderful support. All right, then without further ado, I would welcome our first presenter. Our first presenter is Dr. Wei Kai. Dr. Wei Kai is actually a associate professor in Xinjiang University, learning on the evolution, genome, and also the wild applicability of the wild plants.
Good morning, everyone. I am Wei Kai from Xinjiang University. First of all, I'd like to thank the SSE for hosting this symposium for Mandarin Chinese speakers, and I'm very honored to be invited to have this opportunity to share my work with you. My topic today is the genetic basis of adaptive evolution in wild tomato Solanum chilense. And my presentation will be divided into 3 sections. The first is the demographic history for Solanum chilense, and next the colonization and time of new habitats, and thirdly, how copy number variables affect the diversity of the gene structure. Before I begin, I'd like to introduce how tomato starts from being a decorative plant to foods on our table. So it has a very important position in our culinary culture, but earlier it was just a wild plant in South America, very acid, very sour, and it's grown in very dry environment in deserts, and occasionally birds transport the seeds of wild tomatoes to Mexico and then took root, and then the evolution began. The first domesticated tomatoes were Solanum pimpilifolium. It was domesticated in Mexico, in the smaller cherry tomatoes. So we would deem Mexico the first origin of tomatoes domestication. And all the way to the 16th century, Spanish people introduced tomatoes from Latin America to Europe. But at that time, the plant was deemed toxic and only decorative were shown as planted in the gardens and not enjoyed on the table. And then tomatoes were transported from Europe to North America. It's only be seen as a food crop since mid-18th century, seen as a fruit or a food. And in late 19th century, tomatoes are widely grown in in North America, so it spread to a larger scale of farming. So and then the species or the varieties are introduced back into Europe once again. And afterwards, tomatoes are also introduced to Southeast Asia, and that was in Ming Dynasty. And as the genomes of tomato were published in 2012, the development and domestication and the development of tomatoes have been growing rapidly, and we know more about the domestication process or journey of tomatoes. And I have to mention one scholar, Dr. Charles M. Mick from University of California. He's one of the first pioneer in preserving the wild genomes of tomatoes. And he has a collection of those seeds, and he also distributes the seeds to researchers across the world. And currently, there are 16 species of wild tomatoes, and there are more than 1,000 natural habitats and the coastal Southern America. From coastal areas, the deserts, the way to in the mountains, the Andes Mountains with high altitudes. And our subject, Solanum chilense, spreads across southern Peru and northern Chile in extreme dry environment. Their ancestors were probably spread from southern Peru in the deserts or at the rim of the deserts in Peru. And according to research, during different times, the species colonized in the Akatama Desert. In the earlier stages, they colonized the coastal areas, and the later colonization took place in the Andes Mountains. And we learn that in my research, we found 7 different populations, and we took 35 samples. And among these 7 populations, they are located in 3 different types of habitats. The first is central. So, and then with southern highlands and southern coastal areas. So from the climate speaking, southern highlands and central highlands are very different. But the coastal areas and the central areas are more similar. From previous research, we know that the habitats, the diversity of these habitats is high. And from several candidate genomes, we found that there is a high diversity in terms of genetic diversity as well. That's why we select this species for our studies. So first of all, from the 35 species populations, we established the past demographic model and decolonization events. And we categorize all the samples into four different groups, including the group of central populations and southern highlands, and two groups of southern coastal populations. In particular, for the southern highland populations and the central populations, there are several differences, and we analyzed the demography of all these different populations, and we found that the central populations have the largest populations, and then southern highland, and then coastal populations. So in the last glacier period, there is a high level of specification, and the populations of the southern coastal and highland populations decreased. And after the ice age ended, the populations grew. In the latest periods, the southern populations grew and expanded greatly. And with cross-references and analyzing the migration list, we constructed this demography model, which includes the changes in size of these populations and the time of differentiation and the flow of migration. So from the ancestral populations, we found that the central populations differentiated first, and then the southern coastal populations. But those also took place at a very early stage. And the populations for southern highland populations differentiated at the latest date. And we cross-reference the climate data of the past, and now we learn that the species ecological niche span from coastal areas to the mountains to the higher altitudes. And the coastal populations decreased extremely, and only two populations remained. So we found that climate has a huge impact on the changes in populations. And these are the indicators, including single nucleotide polymorphism, and other indicators are included in our model. Lastly, according to the history, we identified two colonization events southward. One is in the coastal area. The other is colonization across the Andes Mountains southwards. However, the number and size of these populations in the coastal areas have reduced greatly, and the populations in the mountains are growing very fast. And that the result is consistent with our estimations using NLR. In these two events, which genomes are relevant to their habitats, we conducted selective sweeps to identify the positive genes relative to positive selection. We use two approaches. First is using SFS. Second, we use omega plus. And according to our demographic model, we ran more than 10,000 simulations based on neutral data, and we swept and selected the data indicators for positive selection. And only through these simulations and two different types of selective sweeps, we identified all the genes, the candidate genes that are effective in positive selection. And we came up with 799 candidate genes, and we identified the times of those genes were selected, and the results we come up with are consistent with our assumption of the time of colonization. And we looked into the gene ontology of these candidate genes, and we found they're relevant to the response to environmental changes and to metabolism. And we identified four candidate genes, including circadian rhythm, photoperiodism, and vernalization response and protein lipidation. Especially for vernalization and protein lipidation, these two are responses to lower temperatures and to droughts. Root hair cell differentiation is related to droughts. So these genes indicate the difference between the populations in the mountains and the coastal areas. And we established a network, adding all these candidate genes to the regulatory network. And we focus on flowering time. And the networks included fertilization and regulatory network for flowering. To understand the association between the genotype and environment, we conducted redundancy analysis to establish the correlation between genetic evolution and climate change. And we found that temperatures, respiration, and solar radiation are relevant indicators. And then targeting the four genotypes I just mentioned, that's redundancy analysis on these as well, and the RDA results are different. For example, the photoperiod flowering and vernalization response are both relevant to the period time of flowering, but they are responsive to different indicators, including temperatures and drought. And the RDA result of root hair cell differentiation is also different. So from all these analysis results, we identified some mutations that can be verified later. So the age of selective sweeps and timing of selection. From the studies, we found that for the coastal populations, the time are more sporadic. That may be because they are of the earlier times. And we estimate that the populations encounter multiple selection periods. And the populations of the highlands area in the mountains, the times or the age are more concentrated at the end of the last ice age. And we had another a study on drought. We learned that drought-related genotypes, for the older positive selection genes, are at the center of these regulatory networks. They have more commonality and they are more severe. And for the younger positive selection genotypes are at the peripheral of the networks because they have less commonality and connectivity. But that is a preliminary discovery. We need to further discuss or study how it is going to be relevant to our research. So in summary, we learned that Solanum chilense, we reconstructed the species differentiation and colonization events of the species, and we built the demographic model, and that provides a neutral simulation and the parameters and the basis of theory. And the selective sweeps refer to the adaptation to different living environments. And in specific regulatory gene networks, we learned that the positive selection events only come up or occur in specific time. Next, I want to talk about copy number variables and how it shapes the diversity of genotype structure. CNV is a structural variation. It refers to a repetitive or missing piece on a DNA strand. So it could be, this is duplication, gain of DNA. Another is deletion, the loss of DNA. And CNV exists in all organisms and plays a pivotal role in evolution and adaptation. CNV also reflects, the changes in CNV also reflects the historical events in a population's evolution, including selection, reconstruction, and migration. They would affect CNV. So we incorporated 4 different approaches to look at CNVs. And we came up with more than 210,000 CNVs. So including duplication and deletion. And looking, the number of deletions are 3 times of the duplication. However, the length of duplication is much longer than deletion sections. In deletion, 56% are the ones lower than 1,000, 1,000 pp, and that's much lower than duplication. And the sections are not normally distributed or homogeneously distributed across the DNA. And we looked into CNV's results, and the most are the ones with intergenic variations, and a lot are in 5 kb upstream, indicating that CNV affects the functions and expressions of genes. We later analyze CNV and the structure of the populations. And the results show the results are consistent with our SMP analysis. And CNV is used to calculate it in how FST and SMP evaluation. And from this graph, we can see that so the FST based on SMP is highly correlated with VST, showing that we can capture the structure and differentiation of the species using these methods. And are these differentiation relevant to climate? And we first looked at VST values. So we conducted 1,000 limitation tests for each gene. For each, and in the test we defined the differentiation, the genes with more than 95% of differentiation is called differentiated gene, and the ones, and we've located, or we identified more than 3,539 differentiated genes and 2,192 extremely differentiated genes. Then we conducted research on the genes relevant to differentiation. And that's we learned that all these 7 different populations are separated into 4 different clusters. So for the candidate genes of CNV, the results show that all these genes are relevant to metabolism and reproduction and response to the environment. We found 400 genes are responsive to stimulation of the external environment, including sensitive to light, heat, solar radiation, drought, low temperature, etc. All these candidate genes also affect regulation of flowering, specifically photoperiod and flowering are very relevant, and these results are also consistent with our SNP analysis, and we determined that this tomato species is responsive to the pressure of lights and droughts, and they are very relevant to the flowering period. And I will not go into the details here. We selected several key genes and to look at the variation in copy number in the 2 flowering routes, including photoperiod and vernalization. Copy number increased in the southern highland populations. For the coastal populations, there are a lot of deletion and loss of DNA. In terms of root developments, the southern populations all experience deletion. To have a more comprehensive look on the changes of CNV, we conducted the contraction and expansion analysis on copy number. In general, the inland populations have a high level of expansion. If you look at the red square of the numbers, if the number is negative, shows that the copy number contracted. And if the number is a positive value, it shows expansion. For the coastal populations, the copy number contracted. For on land populations, expanded, especially in the highlands. For individual populations, southern highland populations both indicated a high level of expansion, and the coastal populations in the south showed the opposite situation, especially for LA 4107, the population expanded, and LA 2932 contracted. And if we look at the pie charts on the map, you can see that the 2 routes of colonization show different numbers. The route from the Andes Mountains, the copy number expanded. And the coastal routes, the copy number contracted before expanded again. The more interesting thing is that the two populations of the southern coastal populations are at the earlier stage. However, during the selective sweep, we didn't identify any differentiation for these populations. But if we look at the copy number variable, we found an opposite result. And we identified 16 genes that are changing rapidly. So that's an opposite result to our previous analysis as well. So this shows the 16 genes, their change in the coastal populations that has more to do with photosynthesis. And compared to other populations, the genes also exist in other populations as well. In central populations and the in particular population that has a rapid or extreme contraction. And for LA4107, it's rapidly expanding. So this change in CMV is also critical, especially when we cannot explain the pie charts or the dots that we see on the map. We can look at structural change to see if we can find any interpretation or explanation for this situation. And then we conducted a correlation study of CNV and environmental indicators. First, we conducted RDA analysis on all genes. There's no significant results for any genes. And we selected the genes with VST larger than over 0. And we found 3 genes. But for the inland populations, all the genes are all centered, so only, and when we look at other differentiated genes, we found 6 overrepresented climate variables, and the distribution, and it's consistent with distribution of the populations. So that explains that more than 50, and that also explained over 50% of CN differentiation. And for graph C, it shows the 6 climate variables and how the percentage of how they can explain CN differentiation. And this shows the relevance of populations and CN differentiation. And I will not go into the details due to time constraints. So from RDA analysis, we identified the 6 environment indicators, and we cross-reference the copy number of the genes and these elements to conduct an LFM analysis. And we had 312. We found 312 candidate genes relevant to environmental change. And the most interesting or worth noting is the genes relevant to solar radiation, because the gene is responsive to ultraviolet lights. So all the genes in the populations of higher altitudes have increased. And we found that the genes of synthesizing this chemical has mutated. So the tomatoes here cannot naturally synthesize the chemical. And so if you see any purple tomatoes in the market, they are artificially cultivated. However, in the natural populations in the mountainous areas, the tomatoes, because of the genes mutation, so the tomatoes there can grow anthocyanidin naturally. So you can find purple tomatoes there. And so that's a natural heritage or resource of natural purple tomatoes. So here's the second part of the summary. First, Solanum chilense has abundant copy number variations, and CMV can also capture the historical development of the species and populations. And CMV is relevant to differentiation of the populations and reflect the evolutionary history. And in the southward expansion, natural selection may be responsible for how CMV responds to climate change. So in summary, we try to establish the evolutionary history of this species and how they adapt to the new habitats. And so we can have a more abundant resources in terms of species cultivation. So going forward, we will look at the integration of different genomics, including the integration of metabolomics, transcriptomics, and epigenomics. And we need to verify the functions of different genes. That's very important. And that's the research are conducted by my collaborators and partners. And I'd like to thank my team at the University of Munich and the funding support. Thank you very much. This wraps up my presentation. Thank you. Thank you, Dr. Wei, to be our first speaker, sharing a lot of interesting knowledge on tomatoes. Next, let's invite our audience members to ask questions at the Q&A panel, or you can also raise your hand, click the raise your hand icon, and we will invite you to turn on the microphone and to ask the questions directly. I have a lot that I presented, so I didn't go into a lot of details. Pardon me on that. So do we have any questions from our audience?
那就先再一次谢谢我们的魏教授。
Okay, then once again, thank you, Dr. Kai Wei. And all of our audience members, if you have any question for any of our speakers, feel free to use the Q&A section. And we will now take a 5-minute break. So we will come back at, we now have a 10-minute break. So if you have any questions, feel free to ask them in the Q&A section. So we will come back at 10:50, and the session will resume on 10:50 China time. Thank you.